Hello everybody, a very good morning to all of you. Today, welcome to another video tutorial from my channel that is about the recombinant DNA technology. In this video, we will discuss about the first part of recombinant DNA technology. Okay, let's know what is recombinant DNA and how it was developed, on which basis it was developed, and what are the procedures of preparing recombinant DNA. This present day technology was discovered after the experiment conducted by Cohn and Bayer in 1970. In 1970, Cohn and Bayer they conducted experiments in Salmonella typhimurium. Okay, Cohn and Bayer for the first time. Cohn and Bayer for the first time they successfully implanted plasmid of Salmonella typhimurium. It was a bacteria which was first manipulated by Cohn and Bayer. After this experiment of Cohn and Bayer, the recombinant DNA technology have been developed. Okay, this technique involved manipulation of genes. It involved manipulation of gene. The advancement after this technique able to combine two different DNA with the development of molecular technology, it is now possible to isolate and clone a specific segment of DNA, and it can be introduced into another DNA. Okay, so let's know first what is recombinant DNA. Suppose after this experiment of Cohn and Bayer, the concept of recombinant DNA technology has been developed, and it also consider the isolation, cloning of DNA. So what do mean? Let's first know what is a recombinant DNA. Suppose there are two DNA. This is one DNA. This is one DNA with certain advanced feature. It carries some genes with specific advanced feature. Suppose it is another DNA which need modification. This DNA has desired or gene of interest, but this DNA need modification. The DNA that is to be modified is called as host DNA, and other DNA which became the source of gene of interest is called as source DNA, or it is otherwise called as foreign DNA. In recent time, it is possible to isolate a segment of DNA from the source, and it can be introduced into host. Suppose we have isolated a small segment from it. Suppose a small segment has been isolated from it, which carries the desired genes or gene of interest. Suppose this is the segment. This segment is transported to host by means of a vector. Vector is one of the carrier molecule which transport the segment to the host. Okay. When vector receive this gene of interest, it get modified. What happens after modification of host DNA? After modification of this host DNA, a different type of DNA has been produced. This new DNA carries the gene of interest. That means the desired gene successfully implanted into it. Okay, this is a new type of DNA produced after combination of source DNA and host DNA. So it is called as recombinant DNA. This DNA is called as recombinant DNA. The process by which the recombinant DNA is produced is called as recombinant DNA technology or genetic engineering. Okay. So, in recombinant DNA technology, we require two DNA, and by the help of advanced molecular technology, both of them combine to produce a recombinant DNA. It is the process. It is the DNA produced after recombination is called as recombinant DNA. The process in which recombinant DNA are produced called as recombinant DNA technology. Clear? Okay. So, what are the tools, basic tools required for the purpose? Okay. From this we can easily analyze. We require two DNA. One is host DNA and another is source DNA. We require two DNA first. Then the second one is for cutting the DNA. We need molecular scissor, that is called as restriction endonuclease enzyme. Then the third one is a vector which will transport this segment to the host, that is a vector to carry this segment. Then after vector there is a molecular glue for combining the vector and the fragment DNA or gene of interest, that is the DNA ligase enzyme. This DNA ligase enzyme, these are the molecular tools of recombinant DNA technology. Clear? So, for preparing recombinant DNA, we need these all. Okay. When all these things are available to us, we can easily prepare the recombinant DNA. Okay. Then let's discuss about different steps involved in the process of recombinant DNA. Okay. The first step is selection and isolation of DNA. Selection and isolation of DNA, which is almost essential. Selection, isolation of DNA. Okay. Then how to select DNA? As you know, on the view of morphological expression, we can determine the nature of a gene. Okay. Certain organisms with desirable expressions are taken as the source, while some individuals, those need modification according to our desire, are called as host. So, there is no specific method for selecting source and host. It is based upon the phenotypic expression of organism. Okay. So, after selecting cells, how to recover DNA from it? We will discuss today about the isolation of DNA from a cell. The cells are, when after selecting the cells was treated with NaCl and water, NaCl and water, which make the cells more comfortable to be used for the purpose. It can able to make the internal condition, that means the cellular media can be maintained through it. After treating with NaCl and water, we have to treat with STS, sodium diethyl sulfate. What is its role? It can disrupt the membrane of cells, nuclear membrane, cellular membrane, everything get lysed. So, ultimately we can obtain cell lysate. This cell lysate consists of RNA. This cell lysate consists of RNA, DNA, protein and other cytoplasmic components. But out of these, we have to select DNA only. So, for collecting DNA, we have to disrupt the proteins first. So, the cell lysate is treated with proteinase enzyme. This enzyme can digest the protein. Then what happened? After digestion of protein, it is treated with phenylchloroform. This treatment causes sedimentation of protein. The protein get sedimented. So, after that, the process of centrifugation can isolate the proteins in form of pellets. Okay. After isolation of protein, it is treated with ribonuclease to digest RNA first. When RNA is digested, the rest components carries DNA. So, ethanol or acetate ethanol, we can say acetate ethanol is added, followed by stirring with a glass rod. After stirring, we can easily get a white fibrous precipitation. This precipitation contains DNA. Okay. This is all about the process of isolation of DNA from a cell. In next video, we will discuss about the enzymes, vectors and other mechanisms. Thank you all.